Namaste everyone, I am Dr. Amit and welcome back to my YouTube channel Dr. Amit Official. If you haven't subscribed yet, make sure you hit that subscribe button right now so you don't miss out on high-yield content. So in our last video, we talked about metabolism of pyruvate. Now today we are moving one step ahead to one of the most important cycle of biochemistry, that is Krebs cycle, or we also call it as a TCA cycle. And this is a super high topic for USMLE step 1 exams, postgraduate entrance exam and MBBS exam. So stay focused. I'll make it easy, quick and exam oriented for you. So now let's get started with our today topic, that is TCA cycle. So TCA cycle or Krebs cycle, okay. So while giving the introduction to TCA cycle, the TCA cycle is a final common pathway. Okay, always remember this is a final common pathway of carbohydrate, fat, protein metabolism, and it occurs in mitochondria. Okay, MCQ asked from here that where this TCA cycle occurs. So it occurs in mitochondria. Why? To generate energy. Okay. Now while talking about the flow of this cycle, okay, so we'll talk about this flow chart. Okay, so while talking about the flow of this cycle begins when the 3 carbon compound pyruvate is getting converted to acetyl coenzyme A, as we have already learned this in the previous video of pyruvate metabolism, that how this acetyl coenzyme A is formed, which is a 2 carbon compound by using the enzyme PDN, that is pyruvate dehydrogenase. Okay, now at once if you'll see, okay, how the flow is going. So the citrate is getting converted to isocitrate, then it's get converted to alpha ketoglutarate, then it get converted to succinyl coenzyme A, then it get converted to succinate, then fumarate, then malate and oxaloacetate. Okay, so now we'll talk about all these steps one by one. Okay, so now let's see all the steps. Okay, so we all know that pyruvate is getting converted to acetyl coenzyme. Okay, now this acetyl coenzyme, which is a 2 carbon compound, okay, this oxaloacetate, okay, this oxaloacetate and this acetyl coenzyme A form citrate, which is a 6 carbon compound. Okay, and the enzyme used over here is citrate synthetase. Okay, now this step, okay, this step can be inhibited by ATP. Okay, this here is written minus ATP. Don't take it as that 1 ATP is consuming here. No, this step can be inhibited by ATP. Okay, now while the citrate is formed, okay, now this citrate is getting converted to isocitrate. Okay, so the iso is the same form of the citrate and this is also a 6 carbon compound. Now this isocitrate get converted to alpha ketoglutarate. Okay, so and over here the enzyme used is isocitrate dehydrogenase. Okay, so in this step we get one molecule of carbon dioxide and one NADH. Okay, and this step can also get inhibited by ATP and NADH. Okay, but this step is activated by ADP. Okay, now we can see that this alpha ketoglutarate, which is now a 5 carbon compound. Okay, this isocitrate is 6 carbon, but now this alpha ketoglutarate is 5 carbon because 1 carbon is lost here. Okay, now this alpha ketoglutarate is getting converted to succinyl coenzyme A. Okay, while converting this alpha ketoglutarate to succinyl coenzyme A, we are getting what? 1 molecule of carbon dioxide and 1 NADH. Okay, and this step is also inhibited by NADH and ATP. Okay, the enzyme used over here is alpha ketoglutarate dehydrogenase. Okay, now we'll see that this succinyl coenzyme A is now 4 carbon compound. Why? Because again 1 carbon is lost here. Okay, now this succinyl coenzyme A, which is a 4 carbon compound, is forming succinate, which is also a 4 carbon compound. Okay, and over here we are getting 1 GTP and coenzyme A. Okay, and this is a very crucial step. Okay, most of the time MCQ asked that in which step substrate level phosphorylation occur. Okay, so always remember, while converting succinyl coenzyme A to succinate, substrate level phosphorylation occur over here. Okay, now this succinate, which is a 4 carbon compound, get converted to fumarate. This is also a 4 carbon compound. Okay, and over here we get 1 FADH2. Okay, now this fumarate, which is a 4 carbon compound, is getting converted to malate, which is a 4 carbon compound, and over here we are getting 1 NADH. Okay, and then again this malate get converted to oxaloacetate and the cycle goes on. Okay, so the main thing is we are getting 1 NADH over here, we are getting 1 NADH over here. Okay, and 1 GTP over here, 1 FADH2 over here and 1 NADH over here. Okay, now always the other important thing which you must remember from this step, that all these starred, okay, this asterisk is, these are the irreversible steps, okay. The citrate where the enzyme citrate synthase has been used, isocitrate dehydrogenase and alpha keto dehydrogenase used, okay. So these are the irreversible step. Now which one is a rate limiting step? So all these three steps considered as rate limiting, but if you'll get option in the MCQ, then always take this step, okay. This isocitrate dehydrogenase step is a rate limiting step and this is a rate limiting enzyme, okay. Now in the next page we'll see about how the ATP is getting produced. So before calculating ATP, just remember that the rate limiting or regulatory enzymes are citrate synthase, isocitrate dehydrogenase, which is the most important, and then alpha ketoglutarate dehydrogenase, okay. Now I have already said that over there in the this figure that this ATP, okay, this ATP where we have written that minus, okay. So while talking about the regulation of the Krebs cycle, okay, the Krebs cycle get inhibited by ATP and NADH, okay. Why it inhibit? Because energy is already high in those steps, okay, and the steps of Krebs cycle get activated by ADP and calcium, okay. Now what were the important steps, okay? So while talking about the important steps, okay, remember about the decarboxylation step where the carbon dioxide is released, okay, these two steps, okay. So these are the two steps where the carbon dioxide is released, okay. MCQ asked from these two steps that which in which step decarboxylation occur, okay. So this, so isocitrate to alpha ketoglutarate and alpha ketoglutarate to succinyl coenzyme. Now I have already told you in the chart that substrate level phosphorylation occur when succinyl coenzyme converted to succinate where GTP is formed, okay. Now linking that Krebs cycle to ETC, okay, we all know that TCA cycle itself doesn't directly produce much ATP, okay. So it need electron transport chain, okay, so that the NADH and FADH will enter the electron transport chain and can produce ATP. The next important thing which we must remember that TCA is both catabolic and anabolic cycle. That's why it is also called as amphibolic nature. Now we'll see some clinical correlation, okay. So if there is thiamine deficiency, then that will affect the alpha keto dehydrogenase, okay. So alpha keto dehydrogenase, this enzyme will be affected if there will be thiamine deficiency, okay, and that leads to decrease in ATP. Now the second one is in arsenic poisoning, it inhibits a lipoic acid and block the TCA enzymes, okay. And the third one is hypoxia. We all know that in electron transport chain oxygen is needed. So if there will be hypoxic condition, then ETC will be stopped and NADH accumulates and TCA will slow down, because we all know that this NADH, if NADH will be more, then it will inhibit the cycle, okay. So that's why if NADH will accumulate, TCA cycle gets slow. Now TCA needs, so we can say that the TCA needs oxygen indirectly. So where is exam trap? The exam trap is in RBC there will be no TCA. Why? Because there is no mitochondria and 2 carbon dioxide is released per cycle, okay. Now we'll talk about ATP. Now while this is important part, okay, just focus, okay. While talking about the ATP calculation, we all know from TCA cycle we get 3 NADH, 1 FADH2, 2 CO2 and 1 GTP per acetyl coenzyme A molecule, okay. So we all know 1 glucose will produce 2 pyruvate and 2 pyruvate will produce 2 acetyl coenzyme A, okay. So 1 NADH give 2.5 ATP, 1 FADH2 give 1.5 ATP and 1 GTP is equal to 1 ATP. Now let's calculate. So 3 NADH is equal to 3 into 2.5, that is 7.5 ATP. 1 FADH is equal to 1 into 1.5, that is 1.5 ATP, and 1 GTP is equal to 1 ATP, okay. So total 10 ATP will produce from 1 molecule of acetyl coenzyme A. But we all know that from 1 glucose we get 2 acetyl coenzyme A, okay. So 1 acetyl coenzyme A produce 10 ATP, but 2 acetyl coenzyme A will produce 20 ATP from the TCA. Now I hope that is clear that how 20 ATP is producing from the TCA cycle, okay. Now remember that the glycolysis is occurring in the cytoplasm, that's why NADH need to enter mitochondria to the shuttle for ATP production. But this TCA cycle occurring in mitochondria, so don't need to enter ATC via shuttle, so it directly produces the ATP. So that's all about the Krebs cycle. So if you like this video, please do like, subscribe and share this video. Thank you.